Hi, I'm Heather James, conference producer at Technology Networks, and I'm here to introduce our next talk, AI Spatial Biomarkers and Diagnostics are critical for next-gen therapeutics. Please join me in welcoming Dr. Jason Reeves, Director of Application Science at Nucleai. Dr. Reeves is a bioinformatician and translational researcher focusing on developing novel platforms and methods for identifying biomarkers for disease and therapeutic intervention. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. I will now hand over to Dr. Reeves to begin his presentation.
Thank you so much. And I first want to thank Technology Networks for having us here today. This is a fantastic forum, and you're going to be hearing a lot of wonderful talks about how spatial biology is driving innovation across research and development of drugs. We're going to focus our talk today really on the drug development and biomarker development space, which might be a little bit more nuanced in terms of what we focus on. But I think it really it shows the impact that this field really can drive going forward. And I want to sort of set the stage here knowing that a lot of the talks may be centered around technologies. This is going to really need a little bit of context for drug development. So let's jump in by just talking about why spatial biology really is poised to impact drug development. When we think about where biomarkers have been really adopted the most, one of the biggest areas is oncology. And it's been an ongoing evolution of adoption that has really driven this particular market. First with genomic biomarkers and targeted therapies, a few biomarkers that are spatially resolved in immunotherapies. And now as we start to see adoption of antibody drug conjugates and multispecifics, whether it's T-cell engagers or bispecific engagers, these are areas where the drug modality itself is a spatially resolved drug modality, and therefore it is going to directly need a spatially resolved biomarker with it. Now, what do I mean by this? Let's talk a little bit about each of these drug modalities that has a spatial component and why it's important to have a biomarker that is specific to that spatial orientation within the tissue. First, with IO, the main component that was being considered was whether or not a tumor actually was poised to respond to immunotherapeutic intervention. And this really asks the question of what's the structure of the tumor microenvironment? Are T cells and effector cells that can engage with the tumor included within the tumor itself, inflamed, or are they excluded and surrounding the tissue and unable to actually integrate and impact the tissue itself? As we move to additional therapeutic modalities, antibody drug conjugates and multispecifics have different mechanisms of action, but they're both mechanisms of action that require a spatial component. Antibody drug conjugates have a known effect that's called a bystander effect, where they kill not only antigen-specific cells, but cells surrounding them by delivering the payload to a local microenvironment. And multispecifics are drugs that require direct co-expression of two different targets for them to be activated in many cases. And so all of these different drug modalities are ones that the spatial relationships between individual cells and even the subcellular localization of individual proteins is a critical component to the response to that drug. Now, one thing that we have seen is that this is a theory that can be tested directly within treatment regimens and clinical trials. And we've started to see consistent evidence of the need for these spatial biomarkers popping up again and again. On the left-hand side, what we're showing here is recent data that's been published by AstraZeneca showing the role that spatial biomarkers can have in antibody drug conjugates, where a complex spatial score that looks at the heterogeneity of the tumor is actually very predictive of patient response in these settings. Similarly, with multispecifics, we've been actively partnering with a company to develop a novel AI algorithm to help directly quantify whether or not individual cells are co-expressing two different targets. And this is being utilized in prospective enrollment right now, and we'll have readouts soon in the future. And additionally, we're also seeing advanced spatial platforms like multiplex immunofluorescence start to take off. We've seen this partnership between Akoya and Acrivon, where they're driving a relatively low-plex, multiplex immunofluorescence assay that's looking at the tumor microenvironment to try to understand whether or not patient response is driven by the structure of the tumor itself. And I say all of this because we're seeing evidence starting to pile up that these biomarkers are poised to really help the next wave of drug development. However, what we're seeing is that the next wave of drug development is going to be a very complex landscape. Just like with molecular biomarkers, there's going to be a large number of targets that are potentially in play, and many of them are going to require a spatial biomarker. So all of this really leads to the place where we need to be really efficient and really advanced with how we manage these biomarkers. And this is really one of the places where AI can come into the picture. The reason that we believe that AI is going to be beneficial in this space is because it has two different roles. It can help expand what we can do with existing assays and capture really quantitative interactions between different cell types, as well as trying to understand novel assays and create novel scores that really are impossible for the human eye to calculate on their own. And so we break these types of applications into AI aiding and AI augmentation. AI aiding is going to be doing things like increasing the dynamic range, looking for very low expression, identifying rare cell types, whereas AI augmentation is going to be working with novel scoring systems such as a spatial proximity score or novel assay types or integrating slides that might be part of a clinical trial. Now, we at Nucleai are a company that's geared towards enabling companies, whether it's a biopharmaceutical company or an IVD company, to deploy these types of spatial biomarkers directly within their clinical trial settings. And we believe that this will empower the next wave of diagnostics and treatments that are going to hit the clinic. So I want to talk a little bit about what we offer and how this can impact drug development. So we develop AI algorithms for imaging modalities that are tied to pathological specimens. These can be any type of imaging that's done on pathology specimens that can be either H&E, IHC, or multiplex IF. We pair this with these algorithms with a digital viewer, and we can deploy these throughout application spaces that are both investigative or ones that are geared really towards the clinic. And so whether we're working in a retrospective analysis setting or a prospective analysis setting, we've got all the tools to really support this environment. And we work with pharma use cases across translational all the way through companion diagnostic development. And we do have the first AI prospective trial enrollment assay currently being used in the clinic for oncology. Now, I want to talk a little bit about how the clinical development pipeline fits with these different assays. There's different phases of clinical development, and the further along clinical development, the more you need to lock your assay and the more it needs to be geared towards a specific use within the clinic down the road. In the early stages, what we're looking at is identifying mechanism of action and initial biomarkers. And this is ultimately a space where highly multiplex immunofluorescence and even spatial transcriptomics types of assays are going to be really geared towards understanding this complex interaction between different cell types. As you move further and further down the line, it's important to move to a space where you have an assay that can be deployed clinically and can be engaged with clinically. And so we support this entire pipeline because we feel like it's really important for investigators to have tools that can feed the entire funnel, just like a drug development funnel itself is going to be fed. We believe that there's going to be a biomarker funnel that's going to be in parallel to that drug development funnel. Now, I'm going to talk through each of these areas throughout this talk, and I'm going to start with the first area being up at the top, where we believe that multiplex immunofluorescence has a strong role to play in understanding the complex interactions in play within the tumor microenvironment. And we'll move to some of the later stage assays later on in the talk. So I want to talk about what challenges exist for multiplex immunofluorescence adoption and how we are supporting that directly at Nucleai and ways in which we're thinking about this space developing over time. Now, for multiplex immunofluorescence to be adopted in drug development, there really becomes a question of how is it being used and what is it going to be used for? These are complex assays. They require a lot of validation. They require a lot of work to get them off the ground. And what really becomes important is making sure that you design and validate the assay and build a scalable pipeline that's going to support its use in a way that becomes not just geared towards a single experiment, but trying to build a corpus of data that can drive understanding in a larger context. We also think that it's really important to build a path for these assays to be filtered into biomarker identification and selection, biomarker development, and then clinical decision support. And so I want to talk a little bit about how Nucleai is working in this space. We've developed a multiplex immunofluorescence toolkit that we think helps support all of the needs that get from standing up an assay all the way through analyzing it. We've built a robust pipeline for analyzing this data in an automated fashion that scales across many different components that are critical that will need to be accounted for when you're trying to deploy MIF at scale. We also have been working with companies to make sure that the panels that are created are designed in a way that they are ready for the downstream analysis and interpretation. And this becomes critical because it's not just about visualizing a lot of markers, it's about how are you going to use the data at the end of the day. And we believe that ultimately the insights that you derive from this data are the critical component to be robust and to be translatable to downstream applications. To do this, we built a pipeline and a platform that are completely imaging agnostic. We can work with any of the multiplex immunofluorescence platforms. And we also have been integrating with a number of different labs and CROs. So if you're at a pharma sponsor and you're working with a CRO to develop an assay that you want to deploy, either for retrospective or prospective settings, we will probably have a solution available to you and can easily integrate in those settings. So let me make this a little bit more concrete. Let's talk a little bit about what we built here at Nucleai and some of the ways in which we think it can be used in the future. So when we approached multiplex immunofluorescence, it's a technology that's been growing over the last couple of years, and it's really becoming a very well-established technology at this point. We wanted to build a solution that was generalizable so that you could take it and go from one cancer indication to another, and one that would really capture the diverse information that is present within these samples. To do this, the way that our pipeline works is you take an immunofluorescence image, as is shown here. We capture all the cells that are within the image, and we break these down into tiles that are cropped around the cells and decompose those into the individual channels. These are then fed into a deep learning single channel binary classifier that assesses the probability that any given marker is active within that cell based on the overall intensity, but also the staining pattern that it's been trained against. And this allows us then to do standard operations such as cell typing and phenotypic analysis based on a very large corpus of data that we've annotated over time. And there is a bioarchives about this. If you'd like to learn more, the link is down below. The other thing to note is that this is really geared towards a couple key components. We want this to be accurate and robust so that it makes sure that it captures the cell phenotypes appropriately. And we benchmark this against pathologists to make sure that we're not just building an algorithm that looks like it's capturing structure within a UMAP, but also that it's really capturing the biology that we know is true. Secondarily, we make sure that it's generalizable. As I mentioned, we want you to be able to go from one indication to another. And we've tested this not only in oncology, but also I&I. And then we've also wanted to make sure that this is scalable. We want you to be able to run lots of experiments with high-plex studies, and we'll talk about how all of this works in just a second. So as with any multiplex immunofluorescence channel, it all starts with the image. And what we've built is an automated pipeline that's going to do QC and inference-driven thresholding so that this thresholding within a slide is consistent, and it's consistent across cohorts as well. We then build in robust cell typing so that you can accurately identify cells that have known biology within your tissue, as well as look for potential novel clusters within a given indication. Drive this towards a neighborhood analysis so that you are able to then take individual cells and place them into a context where the structure of the tissue is actually physically captured. And then we finally map this to known biologies within a disease indication, as well as extracting interactions between individual cell types and neighborhoods so that those can be interrogated further. Now, as we do this, we've been benchmarking this pipeline to understand its performance compared to standard methods that are existing within the public domain. And if you're not familiar with F1 scores, you're going to see a few of those right now. Effectively, the F1 score asks how much noise do you get from either false positives or false negatives in your system? And the closer you are to one, the better off you are. Anything above 75% for an F1 score is what we consider sort of our baseline that we target for any of our algorithms that we deploy. And what we're showing here is that in the development of this algorithm, when we've been testing it across different cell types that are functionally important to capture within the tumor microenvironment, the deep learning methodology that we've set up consistently outperforms standard clustering-based methodologies. And part of the reason for this is we are using known biology to drive identification of cell types, but also because we are using not just the intensity that's captured within a segmentation mask, but also the staining pattern and informing whether or not we believe the target is expressed. And those two components really help bring up the accuracy of this particular algorithm. Now, when we talk about scalability was another component that we wanted to make sure that we optimize for. We've tested this now multiple times on multiple different indications where we train on one indication using a set marker panel list, and we try to see how applicable it is in another indication. And some markers will not work in every setting where you translate from one indication to another. They may not be present in one indication, or they may have a slightly different staining pattern profile. But generally speaking, we've found that by building out a robust corpus of data to start with, we now have a very generalizable model that can work in multiple different indications with very high F1 scores and very little confusion between the known cell types that are present within the tissue. And lastly, it's really, really important for us to think about this from this perspective of scale. Multiplex immunofluorescence assays are moving into higher and higher plex, and they capture whole slide images in many of the cases that these technologies are deployed. And we wanted to make sure that our pipeline is built such that it can handle any of these types of data. This is just an example from an INI sample showing you our classification of cells from a more than 35 plex assay that's been run. But generally speaking, what we found is plex is not an issue, tissue size is not an issue, and we believe that this builds the baseline for us to then go out and ask questions across large cohorts of data that are important for understanding the context of a given patient sample, rather than asking it just what's happening within a patient. Now, to this end, having a pipeline and having a toolkit is really important, but we also have been thinking about what are some of the spaces in drug development that need spatial biomarkers that really don't have a lot of assays to work with that are truly subcellularly resolved and capture protein expression. I talked to you earlier about why protein expression was really important for a lot of these drug modalities. So when we approached this problem about a year ago, we engaged with Lunaphore and ProPath, and we developed an initial panel for ADCs and multispecifics to look at what the overall structure of the tumor microenvironment is, but contextualize that with some of the targets that were up and coming at the time. Now, I say that this is an initial panel because there's probably additional targets that would need to be added to this to capture the full panel, the full development landscape at this point in time. But we've tested this now on over 200 samples across multiple different lung, colorectal, and stomach cancers. And I've included normal samples as well to understand whether or not this toolkit could be one that would be beneficial for understanding drug response. Now, when we talk about this, I want to put it a little bit more concretely. We talked about why pipeline is important and why it's important to have accuracy in terms of cell typing. But if we move to the actual patient sample, it's not just about capturing an individual cell type and where it lives. It's about contextualizing that to what types of drugs might be appropriate for that patient. And that becomes questions of things like how often do you see co-expression of multiple different targets? These are ones where you want to see both co-expression and potentially co-localization on the cell membrane. Having both components captured is really critical to saying that this is something that could actually be developed as a multi-specific. Similarly, looking for tumor heterogeneity is a common question that comes up. And because we are capturing both the overall structure of the tumor, the localization of individual targets, and the interplay between these targets, we can rationally design potential combinations that might be required to treat the overall tumor architecture in play within that patient. And then lastly, many drugs are ones that you would want to make sure that are restricted to a specific subset of the tumor so that you don't expose a patient to increased risk in terms of toxicities. And the benefit of these assays is you can look for targets that are really more centered in the tumor core and ones that might be potential for antibody drug conjugate development because they have a low therapeutic risk in off-target cell types. And if we take a whole step back, what we view is that the platform that Nucleai has built, both in terms of our understanding of building panels and the overall structure of a tissue, as well as the robust pipeline that we've got underlying this platform, we believe that this can have impacts across the entire drug development portfolio. This can mean things like doing portfolio-wide prevalence studies where you're looking at the balances and trade-offs you have between different drug target candidates, whether it's looking at potential target modalities and identifying what are the right ways of developing a target into a specific drug and making sure that the modality is tailored to that, or whether or not it's appropriate to find specific combinations because we can capture the interaction between the tumor and the microenvironment itself. And then lastly, this also, like I mentioned briefly, does allow us to assess the suitability for development and risk profile that any given drug has. However, all of this is in the multiplex immunofluorescence space, and we do expect that high-plex assays form a critical component for understanding the development of drugs in early stages. But as you move into later stages, we do need to think about how these can be converted into assays that are clinically relevant assays or can be clinically deployed. And so I'm going to focus the next section of the talk a little bit around this area, and I'm going to focus it around antibody drug conjugates just to keep it into one specific modality. But I want to talk about the ways in which standard pathological assays, such as immunohistochemistry or H&Es, can be deployed using the toolkits that Nucleai has developed. So before we get there, I want to talk just briefly about the mechanisms of action that antibody drug conjugates leverage. One of the ways in which antibody drug conjugates work is you have an antibody. It's directly attached to a cytotoxic payload, and it's got a linker that allows that payload to be delivered when it reaches its target. Usually these drugs are antibodies that are internalized, and the linker falls apart due to degradation within the cell. This will cause apoptosis of the original cell that was binding, but will also have a bystander effect that we talked about earlier. And the other component of the mechanism of action may actually be mediated through effector cell-mediated ADCC or engagement and recruitment of those effector cells. And so to capture all this, the benefit that we have is that pathological slides are really rich sources of information, even when they're not multiplexed in a high degree. And we can model a large number of these mechanisms of action directly within these slides. So let's talk about how we do that, or what has been shown in this space. First, one of the first examples where it was becoming apparent that capturing the spatial heterogeneity of tissue and capturing the spatial orientation was going to be important was with HER2, where in HER2 was shown to be effective in an ultra low setting, where previous drugs had not been effective. And this is one where you can score this with a pathological slide. And more recently, in the case of development of Dato-DXd, what we've seen is that a spatial drug score, where you can actually characterize the heterogeneity of the tissue, has been important for understanding the actual efficacy of patients. Well, let's talk through all the ways in which AI can play a role here. One of the things to note about the HER2 low setting is that it's a setting where pathologists haven't really been trained to effectively score HER2 low. If we look at this recently published article, it compares the pathological concordance between multiple reviewers of HER2. And pathologists have been trained to score HER2 at the 3+ level. And so there's very little discordance at this level, but when you move to lower and lower levels, you start to see more and more discordance. This is actually an ideal situation for AI because we can directly quantify the number of positive cells, we can train models for very low expression, and we can quantify directly the number of cells that are positive at a given threshold. Another mechanism that's really been critical for ADCs has been around drug internalization. And this is again a spatial problem. What it's a spatial problem, though, is not necessarily about individual patterns throughout the tissue and how the tissue is oriented, but rather whether or not the staining happens on the membrane or within the cytoplasm. And this is again a setting where AI plays a great role because we can use either computer vision approaches or pathologist-driven approaches to classify the staining that happens within a tissue. And what this means is that we can then take in diverse staining patterns that are either membranal or within the cytosol itself. We can ask the question, does the AI allow you to build a model that can capture these diverse phenotypes of membrane, cytoplasm, or combined expression? And what's that mean? You know, if we're going to have a biomarker that can move into the clinic that measures a relationship between cytoplasmic and membrane expression, it has to be robust. And one of the things that we found when we compare a test where we allow pathologists to score membrane and cytoplasm expression with or without an AI model is that cytoplasmic expression is something that they actually have a lot of trouble with. And so if you're doing an interaction score between the membrane and the cytoplasm, one of the areas that you're actually comparing is going to have a lot of noise. The cytoplasmic component to that specific score is one that really benefits from having an AI model that can be used to aid and train a pathologist as they're moving forward. Now, the last component around tumor intrinsic signaling that has been really important for understanding patient response within ADCs has been around thinking about the overall tissue architecture. And so there's been a rise of scores that look not just at tumor positivity, but look at the structure of the tumor itself. And these are effectively asking the question of, if a drug is delivered to an antigen-positive target cell, how many cells around that target cell are going to be impacted? And we can ask for any given cell that's not antigen positive, is it going to actually see the drug that's delivered to the tumor? So in this setting, for example, if we just scored how many cells are positive within that tumor, this particular sample would only have 16% of the cells positive. Those are the dark red dots here. All the red dots are ones where we don't see significant deposition of that particular antigen. But what we can see is that for any given cell, they may be individually negative, but as you grow further and further out, they actually start to be exposed more and more to antigen positive cells. So if a drug is being delivered here and it has a distribution of 80 microns, it will actually hit this target cell. And so this actually changes the tumor profile from being a 16% positive cell profile to one where you have 27 to 65% of the cells are actually being exposed to the drug within this patient. Now, what this means is that there's potentially patients that by standard assays are going to actually be left on the table. They may not qualify for a given engagement with a given trial. But by using a spatial proximity score, you can actually increase the enrollment that might be necessary. Now, you could also make the argument in this case that you could just move the threshold for tumor positivity down. That's because the relationship between this particular spatial proximity score and the overall tumor positivity is relatively linear. But this is probably the most simplistic version, where you're only requiring one neighboring cell within 80 microns at any given intensity. The benefit of a digital pathology and AI approach here is that you can parameterize this across multiple different types of thresholds to match the mechanism of action of the drug that's known. And that means that we can tune the number of positive cells, the radius, and the intensity. And this gives a much more complex picture of what the drug landscape might actually be within this tumor. You have tumors that are highly homogeneous, where positivity is directly related to the overall spatial proximity score. You also have tumors that are clustered, where there's specific lobes of the tumor that are positive, or it's positive only within the periphery or within the tumor core itself. And so the drug isn't actually reaching as much of the tumor as you might think just based on tumor positivity. And then there's also settings where we see diffuse infiltration, where there's actually small numbers of cells that are migrating away from the tumor nest. These are micrometastases or infiltration. And these might actually have a different response profile than the highly homogeneous tumors, simply due to what's happening as a dynamic specifically within that tumor. Now, in addition to target cell mediated effects, there's also effector cell mediated effects that we can capture with these standard histopathology assays. Most clinical trials are going to have an on-target assay where you're looking for direct expression of your target antigen, but they're also probably going to have some exploratory assays and/or an H&E that can help contextualize this result further. And so you may be able to use a situation like a virtual multiplex. And I'll talk to you about why contextualizing these results together is actually really powerful. So on the left here is an H&E, and on the right is a co-registered IHC. And so you can directly see some of the differences immediately from this. The IHC is going to be really good at showing you where your positive cells are, but H&Es capture a lot more structure. They tell you a lot more about the different cell types. And so within IHC, we might be able to capture a few cell types, but we're going to be focusing on intensity. Within the H&E, we can really get a lot more granular and can ask questions about specific cell types, such as lymphocytes. And this becomes really important when understanding things like interactions and combination therapies. So for example, here, what we're showing on the right is the H&E, but on the left is the IHC that's directly related to it. And we're projecting onto that IHC hotspots of lymphocytes. Now, it's important to show this from a pathological perspective, but what this really allows us to do is now ask quantitative questions of, are lymphocytes trafficking near to the tumor cells that are positive, or are they excluded from the microenvironment? And so we can ask within the H&E, in the regions of the tumor microenvironment that are directly adjacent to the tumor stromal interface, what's the lymphocyte fraction? How are they polarized? What's the density of those cells within that area? And what we found in this particular example was that for antigen positive cells or tumors, where a high degree of the tumors were positive, they also were highly infiltrated within the adjacent TME. And so these are potentially settings where this particular antigen is immunogenic itself, and it might be poised for a combination with the immuno-oncology therapeutic. Now, this is just an example. This type of pattern isn't always observed, but I wanted to show this because it's illustrative of the type of data that you can get with this type of marrying these two types of slides. Now, lastly, sometimes you need co-localization directly, and multiplex chromogenic assays are directly deployable in clinical settings, and they're really great because they can capture multiplexed information. They don't reach the same plex as multiplex immunofluorescence, but they're harder for pathologists to read. And so we built a suite of tools here at Nucleai to help pathologists read these and to build models that can build off top of these types of assays. So in this case, we're showing a virtual dab where we're decomposing the color channels into something that is more akin to what a pathologist is traditionally reading so that they can score each of the channels separately. We also build models where we can take this and ask the question of, do we see specific cell types that are co-expressing individual components within this multiplex chromogenic assay, just like we would do with a multiplex immunofluorescence. Now, all of these AI algorithms and tools that we've talked about now, up until this point, we've talked about them from the perspective of how do they impact our understanding of drugs, right? There's a big sort of elephant in the room here that we also need to talk about, which is how do we make sure that AI algorithms and spatial assays become clinically ready? And this is a component that involves us thinking not necessarily about the science of spatial biology, but about the regulatory framework that they live in and the types of assays that can impact clinical development. And so for us to really be in a situation where our algorithms that we deploy are ready for clinical development, it means that we need to be ready to integrate across multiple different sites, both from the pharmaceuticals company to the clinical trial site to the in vitro diagnostics and CROs that are actually running the tests. And we need to be building our algorithms in a way that they're ready for regulatory agency review. All of this means that we need to have robust data management, chain of custody, and quality management throughout the life cycle of the clinical trial. And so at Nucleai, we don't just develop AI algorithms, but we also build out services that are really geared towards making sure that our algorithms, when needed, are ready for that regulatory environment. We built out a robust QMS and regulatory framework, and we have been partnering with IVD companies on making sure that the AI algorithm is incorporated and tested and validated during the assay development itself. And so this means that we also needed a partnership environment that incorporates multiple different labs and CROs, as well as IVD partners that we can potentially work with as a pharmaceutical company is developing their drug. From a contextual framework within a trial, what this means is that we also need to be ready to integrate with the trial workflow. And what this means is that within a trial, when the trial is going on, we need to be able to robustly and quickly make sure that when a sample comes to a CLIA lab and is stained and imaged, that our algorithm is locked and ready to be deployed in a quick fashion that's not going to interrupt the clinical workflow. What we have built so far has been able to make sure that our algorithms are deployed within less than a day and sent back to the CLIA testing site and then integrated with the pharma and the clinical labs, or clinical testing sites themselves. We directly interact with the IMS of the CLIA lab, the image management service, and then we also work to make sure that the score that we're capturing here is reduced down to something that can be reviewed directly by a pathologist and approved by them, so that when they're working within their workflow, that they have the final say on whether or not the algorithm was correct, and that it's not such a complex interaction or report that they have to spend a large amount of time on it. We want to make sure it's really geared towards the intent of the site to make the decision of whether or not the patient receives the therapy. And so with this in mind, I want to just take this all the way back to some of the framing that I had at the very beginning here. We believe that the spatial biomarker market is growing and evolving in a way that's going to require complex assays. This is because these are drugs that have modalities that are driven directly by the spatial orientation of the tissue, and because we're going to need to start getting more and more out of these tissues without compromising and without exhausting these tissues quickly. If even half of the drugs that are being developed right now are going to require a spatial biomarker, this actually presents a clinical problem in that a biopsy from a patient usually gets you somewhere between 5 to 15 slides. And with the need for also testing molecular biomarkers, we're going to need to proactively address that. So whether that's directly with leveraging the information from H&Es, whether that's leveraging the information from IHCs that are already in routine practice like HER2 or PD-L1, or whether that's deploying a complex assay such as multiplex IF as a screening assay, we believe that spatial is going to directly impact patient care in the not too distant future. And so what I'll leave you with is that we want to be a partner in this ecosystem. We believe that it's really critical for pharmaceutical companies, medical centers, IVD developers, and algorithm developers to all work in concert. Because at the end of the day, the really important thing to remember is that we're trying to make sure that a patient is going to respond to the therapy that they're given and that we get the right therapies to them across the board. And so with that, I hope that I've given you a little bit of perspective on where drug development is going, how spatial biology can impact it, and how we at Nucleai hope to become a partner with you in this evolution of this landscape. So with that, I'm going to stop here and thank you again for attending the talk.
Thank you so much, Dr. Reeves, for that fascinating presentation. I'm sure that everyone will agree that that was really interesting, and thank you so much for coming on to share your expertise with us. I'm sure that we'll have lots of questions, so I'm going to hop straight into the Q&A now, and I've got the first question for you here, which is, do you have any requirements for image format or resolution when developing a model?
It's a great question, and there's a bit to unpack in that. But at the highest level, we work across image modalities and across image formats, as long as it's a standard image format that's supported across pathology, such as OME TIFFs. I want to mention specifically, though, that the resolution part of the question depends entirely on what you're trying to model. We work with 20x or 40x slides frequently, and a lot of the MIF imagings has their own specific resolution formats in terms of max image format, image resolution. But it really depends on what type of feature you're trying to annotate, what you're trying to model. For most features such as tumor positivity, 20x slides are going to be fine. For most H&E models, 20x slides are going to be fine as well. But if you're trying to deep learn something that depends on really crisp outlines of cells or specific features within a nucleus or subcompartment of a tissue or cell, you might want to be working off of a slightly higher resolution. We do want to make sure that all of the models that we develop are also built in a way that they are built towards the resolution and the image format that you're going to be working with on the other side. So you wouldn't want to build a model off of a 40x image if you're only going to have access to 20x images down the road, for example.
Excellent. Thank you for such a comprehensive answer there. Another question that we've had come in that's excellent here is, when using a model for trial deployment, are there any study design considerations that you consider for AI model development?
Right. And so a lot of this question really drives to what are the intended use? If this is a retrospective only trial where you're going to effectively ask the question of, is the model predictive, but it's not going to be used for prospective enrollment, it's not a decision-making biomarker yet, and you're building towards that space, what you're probably going to have is a lower regulatory environment. You're going to have less model optimization for intent to treat populations. And you're going to be really focused around the model performance across the variables that you know that you're going to see in the lab itself. When you're thinking about deploying it in the prospective setting, you're going to have a larger amount of design control that you're going to put in place. You're going to have more samples that you're going to want to have upfront prospectively tested before the model gets used. And you're going to want to make sure that the model itself is built in a way that it can be deployed quickly and easily across the sites, as we mentioned towards the end there. There's also the question of, if you're trying to do an exploratory analysis, trying to balance the type of analysis that you're running with the outcomes, the data that you have available. And so one of the things to keep in mind with any AI model is that the model itself may be robust, but then the picking of which features go into predicting patient outcome should be such that you're following good practices for statistical analysis. You want to make sure that you've got balances between different responder groups. And you want to try to be possible wherever it's possible to have validation available to you, whether that's going to come in the setting of a downstream trial, or if you have other retrospective data that can be used to help validate that the model is predictive.
Excellent, thank you so much. I've got another question that's come in here from Michelle. Are the layers of the image selective? So for example, can you see the image in H&E on its own or H&E combined with IHC, for example?
So right now, the way that we produce our image viewer is to allow you to, we co-register the slides and we allow you to view them side by side. As these are co-registered slides, they do have the same types of features, but we found that when we provide them as side-by-side views, that allows pathologists to compare back and forth between the two readily and makes it very easy for them to work with the data. When we talk about multiplex immunofluorescence or multiplex chromogenic IHC, those are settings where the virtual dabs, the color deconvolution, or individual channels are selective, where you can choose what you're overlaying onto the slide.
Wonderful, thank you so much. And I think we've got time for one or two more questions here. So another great one that I've had is, what are the specific challenges in translating AI-powered spatial biomarkers into clinical practice? And how can you collaborate to ensure robust and reliable clinical decision-making?
Fantastic question there. Obviously, this is an early stage space, right, across the board. Right now, we've been working with pharma partners to deploy these biomarkers in clinical trial testing. The question that you're asking really is about the actual clinical practice side of the house. And part of this is really about building a corpus of evidence that suggests that it can be used, having a place for this test to be deployed, and having a clear, crisp interpretation of the test. Can you repeat the last half of that question for me, Heather? I want to make sure that I fully address it.
Of course, yeah. So asking about how you can collaborate with clinical practice members in order to ensure robust and reliable clinical decision making.
Right. So I think a lot of the work that's being done, it really depends also on the region of the world that you're working in, honestly. In North America, working with major academic centers and focusing on the interactions that can happen at those spaces are going to be where you're going to be able to get the largest interaction space in digital pathology, just due to adoption. Europe, there's a little bit more robust digitization that's been taking place, and so you can potentially deploy in a broader space. And then Asia, there's many countries that are doing full digitization of most of the trial samples that come through and most patient samples that come through for diagnostic purposes. And so you need to think about some of the landscape that you're operating in terms of how this model can potentially be deployed in the future, and whether it would need to either be deployed at the academic site, the research site, or across clinical practice, and whether you'd need potentially a central lab to be a place to do some of the testing. A lot of the companies that are in this space are doing central testing simply because of some of the uptake barriers that exist within digital pathology at this current point in time.
That's great. Thank you so much. I think that we have time for one final question here, and I think this is a really lovely one to end on. And that is, for you personally, what is your most exciting future direction for AI-powered spatial biology?
Oh, that's a fantastic question. I love it because for me personally, this has been a journey watching spatial biology both take off and be a part of that technology evolution. And so I have previously been in companies that have been focusing on technology development and platforms that can measure spatial biology. And I've come over to Nucleai because I really do believe that the next phase is not just understanding the spatial biology that exists within tissues, but moving that into a place where it impacts patient decision-making, patient standard of care. I think it's going to take the community pushing across the board for this to be adopted. And I think that the thing that is most exciting for me to see right now is that there's becoming a robust ecosystem of companies really pushing this line. It's not just one company trying to get the regulatory agencies to understand that there's value here. We are seeing signals both at pharma, we're seeing signals at at individual companies across different imaging modalities. And I really do think that the next 3 to 5 years, we're going to see a growing ecosystem of tests that measure the actual tissue architecture and really understand how that relates to patient outcome in a way that we haven't been able to do until very, very recently. I'm really excited to see what we can learn and project and extract from these images. And I would love to see, and I don't know how much barrier there is to this, but I would love to see the evidence that we're seeing now really drive adoption of digital pathology as a robust and continued use, not just at major research centers, but really across the entire clinical practice space. But that's going to take time, and it's going to take a lot of energy across the board. But I think that we are seeing now the seeds of that getting planted, and I'm really excited to see that shift that's going to potentially happen and really help improve how pathology can drive patient care even further than it already does.
Amazing. Thank you so much, Dr. Reeves, for both your insightful answers and for bringing so much enthusiasm to this space. I know that you are really excited about this, and that definitely comes across in your presentations. Unfortunately, that's all that we have time for today, but we really appreciate you taking the time to share your expertise with us. Any questions that we didn't have time to get to will be sent through to Dr. Reeves after the event. And as with all other talks in the event, this talk will now be available to view whenever you want on the platform for the next 30 days. And that just leaves me to say thank you so much again, Dr. Reeves, and we'll be back tomorrow at 8 a.m. Pacific for day 2. I'll see you there.
Thank you so much. Thank you so much, Heather, as well.
Thank you.